Today we're going to talk about replication. Replication is the first step in gene regulation. Before you can transcribe or translate any DNA, you have to replicate it. The main goal of replication is to create a copy of DNA. Now let's introduce our main players involved in replication. First, we have helicase. The role of helicase is to unzip the DNA helix. An easy way to remember this is that it ends in case, and you unzip a suitcase. Second, we have primase. Primase synthesizes segments called primers that help tell the DNA polymerase where to start its synthesis. Third, we have ligase. Ligase goes along the lagging strand to seal up the Okazaki fragments. Fourth, we have DNA polymerase. DNA polymerase is our hard worker. DNA polymerase works in pairs to create two identical DNA strands from the original DNA molecules. Now, let's get into the actual process of DNA replication. DNA polymerase always synthesizes or writes DNA in the 5' to 3' direction. Here's our DNA strand with the 5' and 3' locations noted. It is split in half on the left side. Helicase is where the replication fork is. It is going along and unwinding the DNA. Another enzyme that is involved in replication is DNA gyrase. Gyrase loosens up the DNA in the supercoil so that it can be unwound and split by the helicase. DNA polymerase works in the leading strand continuously, shown here at the top. It writes in the 5' to 3' direction a new complementary strand of DNA. When it's on the lagging strand, or the strand below, it has to work the opposite way. This is because it has to write in the 5' to 3' direction. It has to go back and start over because the helicase is unwinding in the other direction. These fragments that it creates are Okazaki fragments. Ligase goes along and seals those fragments up. Primase creates primers. There's only one shown here on the leading strand because it only has to tell the DNA polymerase to start at one point. On the bottom, however, it has to continually start over again. That is primase's role in replication. At the end of this, you have two new strands of DNA that are complementary to one another. These can then be used to transcribe and translate for important proteins.